Okay, so as I said, so thanks to the organizer for inviting me, to Damien, to Romain, and it's a great pleasure to be here today. So I'm going to talk, why I can't remove it from the screen. Today I'm going to talk about coding dynamics of the striatal networks during learning. And as Romain said, it's a long story. We start in Marseille labs a long time ago. I will not say how long, and we finished the work because we see it's a combination of different disciplines who participate to this work. And today I will try to introduce this because we have to submit the paper quite soon. So my apologies for the computer scientific which are in front of me. So I am the neurobiologist, so I will attempt to answer to your question, but please don't ask me too much about the algorithm behind. Right, so first of all, I would like to thank my collaborators. So Maxime Villet on the left, who is my PhD student who did the electrophysiology recording on rat in Marseille for this project. Patricia Reynaud-Bourré, which is my collaboratrice in mathematics. She's doing, she's statistician, and she did all the analyses for this story that you will see. And finally, Francesca Sagolini, who is from Marseille at the LNC, and is in this lab that we did all the experience that you're going to see today. Okay, so we're going to enter in the subject. So today I will discuss about the striatum. The striatum is a subcortical region that is part of a basal ganglia, and as you can see on the left, it's in red in human, but we have a very conservative striatum, and we have also in rodents. Basically, in the literature, it has been well described in instrumental conditioning, and so many studies separate two parts of the dorsal striatum. The first one, which is the dorsolateral striatum in yellow, and the dorsomedial striatum in green. And to summarize, I would say that many papers show that the dorsomedial striatum is related to goal-directed behavior in instrumental conditioning, controlled by action outcome contingency. And the dichotomy with the dorsolateral is the dorsolateral is more involved in inflexible motor routine and would be involved also to take the relay of the learning. And when the learning is becoming more an habit, it would be the dorsolateral which taking this process in account. Right, so here it has been well described, as I said, in instrumental conditioning, but what happened in a free learning process? So what do I mean by a free learning process? In instrumental conditioning, all the experiments are precisely time-stamped. So what I mean by time-stamped, so for example, you have a cue, which is a light, which is a song, and as soon as the light arrives, for example, the animal has to press on the lever to receive the food to avoid a food shock. Okay, so this is timestamp. And almost of the experiments where we have done electrophysiology, single unit recording, electrophysiology during behavior, they just use this cue to timestamp and to have a look on the task event related activity of this neuron. Okay, but how can we do with learning without any timestamp? So it does mean that we're going to have a free learning without any cue guide instruction. So today, that I would like to introduce is this behavior that we did. We did single unit recording during a spontaneous alternation task. So to explain more in detail the task, I will show you a little cartoon. So as you can see here, we place the animal at the bottom of the T-maze, and the animals go up to the central stem, and it can choose the right or the left. And so, for example, the first time that it chooses the right here, the example, it receives a reward on the right delivery here. A food pellet, and he's coming back on the other arm, and after this, he has to alternate for the second trial. So if I show you again, because he was quite fast, so go up to the central stem, choose for the first trial, receive a pellet, a food pellet, and go back on the central stem, and now he has to choose the opposite. And we place the animal during 20 minutes, and every day he can do as much as he wants. So it can be 100 trials per day for the 20 minutes. And he has to alternate to receive the food pellet, because if he's doing, for example, the same turn every time, he won't have the food pellet. So that's why it's labeled the spontaneous alternation task. But as you can see here, we don't have any cue to just oblige the animal to start, or to tell him, okay, you have to turn to the left, to the right, or it's time is time on the reward delivery. So during this task that we did, and that Maxime Villet did, my PhD student, is record simultaneously the single unit activity of the DMS or of the DLS. So we implant 4 animals in the DMS and 3 animals in the DLS, okay? And so we were recording the single unit activity, and afterwards we did the spike sorting analysis and all the analyses that I will explain to detect if we have any correlation with the behavior of the animals. So at least we record 293 neurons in 4 DMS rats and 357 neurons in 3 DLS rats. And sorry, we dissociate on the shape of, on the waveform of the spike sorting, the different type of neuron, and we detect 2 types: the medium spiny neuron, which is quite known in the dorsal, in the striatum, and the fast spiking interneuron. And we have 451 medium spiny neuron and 199 fast spiking interneuron, but no difference in terms of proportion in the DLS and DMS. So we pull all these neurons together for the analysis that I'm going to show you next. Okay, so now you know the experiment, and as I told you, it's a spontaneous task, and every day the animal will be trained during 20 minutes, and is going to be during 50 days. So it's a very long, processes. So now we would like to think, okay, how are we going to analyze these results? Because we don't have the timestamp, because we have no cue associated to the reward or to turn left or to start the experiment. So we were thinking, maybe we can discriminate different paths. So, and that I label by path is start from a reward and go to the opposite reward. So for example, if you're looking at on the left, you have 2 correct paths. The first path on the top, so the first is going from 1, go down to A, go up to B, and go to the 2. Okay, so it's onset 1, go down to A, B, and 2. Okay, this is the first correct path. The second correct path is the opposite way. So is I'm starting from 2, go down to A, go up to B, and go to the left to the reward 1. Okay, this is the 2 correct paths, and of course the animal is able to do incorrect path, because we don't force him to go on the right direction. So that means he can go back, go straight, go up, go down. And so actually we identified 10 incorrect paths, as you can see here: a straight path, an opposite path. So we have 10 incorrect paths. Interestingly, if you're looking at on the path under, you have a number. So this is the average time in seconds to do the correct path. And so it's interesting, because if you're looking at the same length of the path, you can see that in the incorrect path, they are longer. So as soon as they know the task, they are going faster to go to do the correct path. I have something in the discussion I continue. All right. Okay, so first.
It's only I know. It's okay.
Okay, so first we represent the data in the following way. I'm going to explain. So on the ordinate, we represent the number of paths per minute, okay? And so you can see the color is associated to the path. So for example, the 2 correct paths are in orange and in red here. And the incorrect paths are the other color here in blue, blue light, blue dark, and the other orange, degraded. And surprisingly, we distinguish 4 stages. I mean, we try to separate 4 stages. So I'm going to explain. The first stage, the animal is doing the same number of correct paths and the same number of incorrect paths, okay? Is the beginning of the learning. Stage 2, we start to have a dissociation. So the correct paths are increasing and the incorrect paths are decreasing. Stage 3, we have a stabilization of the correct path, and we are almost at 90% of success, and we have a decrease of the incorrect path. And stage 4, this is a long time stabilization. As you can see, you have no incorrect path, I mean, very few incorrect paths, but still a variation in the correct path. So keep in mind that we separate our task in 4 stages: beginning of learning, the stage 2, which is the animals start to understand the rule and increasing the number of correct paths. Stage 3, stabilization of the correct path. And stage 4, maybe we become in a routine. So that means we are doing the same number of correct paths. Okay. So now, what are the analyses that we did behind? As I told you at the beginning, is a free learning process, and we have no task event to separate the different behaviors. So that we start to do is to see if with classical analysis that is used in electrophysiology, we could see something in terms of decoding neuron. I will explain later on. So here we use a classical analysis about task event coding neuron. What does it mean? We design different points, strategic points. So the point that we design is 2 onset, the 2 onset, which will be the point 1 and 2. The intersection, we have 2 intersection A and B. And after we have to reward R1 or R2. Okay, so that means at least 6 events. And we cut the time in 500 milliseconds and cut in 5 bin of 100 milliseconds. So here, for example, that we look is the 500 milliseconds before the animals leaving this place, right? So this is before the onset. Here is the 500 milliseconds around the middle intersection box. We're looking at the firing rate around this middle intersection box. And here is the 500 milliseconds after you enter in this box, so where it's supposed to receive the reward, right? So now we have 30 bin because we have 6 events with 5 multiplied by 100 milliseconds, okay? And so we classify the task event coding neuron. Neuron whose firing activity is 100 millisecond interval varied significantly from all over interval. And we used a chi-square detection method. Okay, so as soon as the neuron has a difference between this interval and under the 30 bin, we consider that this neuron is task event coding neuron. Is the definition of the task coding event that we did. And as you can see in the table, you see that we have more coding neuron, task event coding neuron for the DMS, 65.5%, compared to the DLS, where we have only 38%. So it seems that, and this is for all the 4 stages, and we pull the 4 stages together, it seems that the DMS has more task-related events, so task event coding neuron, than the DMS. You see, we have a small percentage of neuron, of non-coding neurons, so we just discard this population. And interestingly, the proportion of coding neuron in DMS is higher than the DLS for stage 1, 2, and 3. But for stage 4, the DLS seems to have more coding neuron than the DMS. So it's going, I mean, it's in accordance with the literature. where they have shown that the DLS probably is taking the relay when the animal is in habit in terms of a learning process, and that's why we could have more task event coding neuron in stage 4. Right. So secondly, that we did with this experiment is have a look on the different stage of the task event neuron. So to do this, we use the Z-score, which is quite common in the type of analysis. So the Z-score is the firing rate of one neuron during the bin of 100 milliseconds minus the average of the firing rate during the 30 bin divided by the standard deviation, right? And we represent the 4 stage. You can see the graph on the left, and we use the average Z-score. So it does mean if you are on 0, okay, you don't have any difference, so it's just a firing rate, a random firing rate, okay? And as soon that is going up or going down, because it can be on the 2 ways, it seems that the firing rate is increasing or decreasing. In dot point, you have the interval confidence. And so if the interval confidence does not cross the line, so it does mean that is significant. So what we can see in these 4 graphs. So I try to summarize briefly that we can see. At the first stage, as you can see here with my the red line, sorry, is around R2, so that means reward. It seems that the DMS and DLS activity is higher because, as you can see, the red line and the blue line are far away from 0. Even if it's under 0, it's still an activity which is different, okay? So we have this activity at stage 1. So according to the literature, and for example, Grebil and colleagues have shown that it's true that the DLS seems to be at action boundary. So that means at the beginning of the task and at the delivery of the task. It's like boarding the 2 task events. So it's in accordance for the DLS. For the DMS is something surprising. Stage 2, we have an increase, especially in the DMS, which is in red, at the B intersection, which is quite interesting and also seems to be in accordance with the literature, because remember, the B is the cross where the animal has to choose between the left and the right. So it does mean that you have to take a decision, so more goal-directed behavior. So the DMS would be involved. No surprise. Okay, for stage 3, nothing really apparent. And for stage 4, it seems that the DMS activity is at action boundary, so similar to DLS. So this is something completely new because no one so far found that the DMS could be also involved in this action boundary. Right, this is the first analysis that we did for task event coding neuron. But we would like to see also, do we have something like a start, a beginning of decoding the network? Okay, I will explain. The entropy is used in the literature, and the entropy is a measure of type of, is used to decode a type of neural code. So the decrease in the entropy, I won't give you the algorithm behind, but the decrease in entropy reflects a decrease in the randomness of the firing rate in the neural population. So it does mean that if you have an increase in the coding ability, you should have a decrease of the entropy, okay? If your entropy is low, it's related to less randomness. So it does mean that probably your network is more coding, right? So we did this type of analysis. So for example, Grebil and colleagues did also the type of analysis. And we use the stage 1 like a reference. So that's why stage 1 is at 0, okay? And we look the entropy for the different stage. And surprisingly, it does not mean that the entropy is related to coding anything, because it's increasing So if it's increasing, the randomness in the network is higher. Except for stage 4, where it seems that you have a decrease in the DLS, but I mean, you have a very big here standard deviation, and it's overlapping and is not coming back at 0 level. So entropy increase between stage 1 and stage 4. Does it mean that the network decreasing the ability of decoding? This is the first question. And the DLS entropy seems to be lower at stage 4 than the DMS. So could mean that the DLS is more coding than DMS in stage 4. Oh, yes.
Is the entropy of the population activity?
Yeah.
Or is the distribution of single neurons in the population?
Okay, distribution of the neurons in
neuron by neuron.
Yeah.
And your entropy is just the absence or presence of spikes. Then in reality, it must be linked to the firing rate.
Yeah, I mean, because it's between 0 and 1.
Yeah.
So
the entropy of 0, 1, what is just the firing rate.
Yes.
It's a kind of more non-linear, but monotonic function. So this basically, is it correct for firing rate or changes? So is the rate of entropy or is absolute?
Okay, so for the entropy, we use the Z-score.
Well, we can
so, but we can discuss after all, because it's true that it's something, I mean, I'm not sure that is a good way to calculate the randomness of this thing, because as you said, it depends on the firing rate. That is the absolute value of the firing rate.
The maximum entropy would be for an intermediate firing rate. Okay.
Exactly.
Neither too low nor too high.
Yeah. So to continue on the type of analysis that we did, the second one is we look at the neuron coding for left or right turn. So what does it mean? Here, we would like to see if we can have a difference in terms of firing rate depending where the animal is going. Okay, so that means if it turns to the right or if it turns to the left. So we compare the firing rate between intersection A and B, and for the correct and incorrect path, and see if we have a difference if the animal is going to the right or to the left. Okay, and we can see that we have a difference higher in DMS again than the DLS, but still 35%. So it seems that the DMS neuron have also spatial direction and could also code this left and right. No difference again between the medium spiking neuron and fast spiking anterior neuron, and we have an increase in DLS for stage 3 and 4. So again, the DLS seems to be more involved at the end of the process when we have a stabilization of the correct path. Right. Another way to analyze was to study the synchronization. Okay. So far, we were looking at firing rate, a Poisson process. So it does mean that we were looking at the firing rate activity of the neuron depending of the task event. Here, we would like to see if pair of neurons during each trial are more synchronized or not. So that Patricia did, is a permutation. So she was doing pair by pair for each section, and by permutation, was looking at if the neuron was synchronized or not. So we calculated the probability of being synchronized at intersection A or B for each pair of DMS and DLS neuron recording simultaneously in each session. So I'm going to try to explain this representation, which is not easy to understand. So here we have a cumulative distribution function of the p-value. So the diagonal here does mean you have no synchronization at all between the pair, right? And further you are from this diagonal, higher than the diagonal, because above the diagonal, we are not sure if we can talk about desynchronization of something else. But if you are higher on the left, I don't know if you see my mouse, yes, but online, I'm not sure. Yeah. So if you're going up here, okay. If you're going up here, it does mean that further you are, more synchronized you are. Okay, and so that she did the cumulative distribution function, but after all, she looked at with KS Kolmogorov-Smirnov statistician test. She has a look if it's different from a diagonal, and that we find is the cumulative distribution function of DMS p-value in stage 2 at intersection B is significantly above the diagonal, as you can see here, right? And in stage 2, so stage 2, you have the different colors, sorry, correspond to the different stage. And on the first column, sorry, I didn't mention, this is the DMS synchronized at intersection A, and under you have the DMS synchronized at intersection B, and on the right is DLS synchronized at intersection A, and DLS synchronized at intersection B. So that we show is in stage 2, we have a DMS p-value, I mean, a CDF of DMS p-value at E and B significantly above the one of the DLS. So what we conclude that the DMS is significantly synchronized and more synchronized than the DLS in stage 2, which is again in relation with the literature when we say that the DMS is involved in the learning process at the time of goal-directed behavior. So stage 2, remember, is the stage where you increase your correct path, your two correct paths, and you slightly decrease your incorrect path. So that means they start to understand the role. Right. So far, what we have? We have seen that the firing neuron can decode. Yeah, you can open it if it's too warm. The firing rate can decode the animal behavior. We have seen that the firing rate task related, we have an activity, and we have seen that the DLS seems to be involved at action boundary, DMS at the intersection section. Sorry. So it seems to be involved for decoding animal behavior. But it's not the only thing, because if we're looking at the synchronization pair by pair, we have seen also that is interesting, the synchronization increasing in the DMS, okay, in stage 2. Right. So we need a model with firing rate and interaction between neurons. And that's why we used a multivariate Hawkes model that I will introduce with my word, that means my neurobiologic word. And so this model will pull together the firing rate and the interaction of the neuron with the directionality of the neuron. I will explain with the graph interaction. So to do this analysis, now we only use the coding full path. What does it mean? We took all the neuron coding for the full path. So it does mean that if a path, one of the 12 paths is the firing rate activity on this path is different from the 11 others, so is coding for this type of path, right? So we still have higher number of coding full path for the DMS than the DLS, and no difference again between the MSN and the 5 spiky internal. Okay, so now I'm going to try to explain what is Hawkes model, a multivariate Hawkes model. Here, it's a graph of interaction. So to explain so far, on the left side, the first column, you have the firing rate activity in hertz. Okay, the color depends on the level of the firing rate activity. The first line corresponds to a graph for one trial and one correct path and an incorrect path. Okay, it's an example because of course we can't represent all of them. It's just to illustrate an example. So the first line is for the DMS and the second line is for the DLS, right? So here classically on the first column, this is a Poisson process, so just the firing rate. And here you have dot node. As I said, I lost my mouse. You have a node here with a number inside. This is the number of your neuron. Okay, so we have neuron 34, 33, 32. And the color will be the spontaneous activity of the neuron. Okay, so the firing rate, the spontaneous activity. So now you pull your neuron in the graph and you have some arrow with a direction. And that is, thanks to the Hawkes model, we can see the excitation between two neurons and the directionality. So what does it mean? So for example, if we are taking this example on the right, you see that between, I can't read from here, but so I think you have 13 here, which is one neuron, and 33 here. And you have this arrow in red. This arrow does mean that when 33 is firing, we're gonna have 2.5 spike in the 33 one, next to the 33. So the 30 exciting the 33 with a rate of 2.5 spike of excitation, okay? And we can put that in this model. So we have a graph for each path, each session, okay? And all the things. So we can't see anything just with the graph of interaction. So that's why we just compare first the difference in terms of decoding power. So how Patricia did. She used the 2/3 of our data to simulate and the last third to estimate. And the decoding power will be the proportion of correctly guessed the path for the animal. So it does mean that whenever the animal is in the maze, thanks to the decoding power, we can predict where it's going, which path it's going to do. Okay, this is the idea behind. Okay, when she has a look on the difference between Poisson process just based on the firing rate, remember, and Hawkes with the interaction between neurons, surprisingly, we can see the Poisson is approximately the same than a random process. So that means that it does not help us to decode anything. And when we compare Hawkes with Poisson process, we see that is the same than compare Hawkes with randomness. So it seems that this model could be helpful to better decode that the neuron code want to say. Okay, so next, I will try to explain. We have a look on each stage, and you can see the box plot here. So we take in consideration in this model different parameters. And the different parameters that we took are the following: the number of paths, the number of neurons, the average firing rate, but also the learning stage, and the comparison DMS-DLS learning stage and the cross effect. So is that the label beta regression and probeating, and we can see that all these parameters are significantly important, and we incremented in the model. So at least what we can see, we see that all the box plots for the DMS are in, I would say peach, yes, orange, so the color of the DMS. You can see that the DMS for all the stage is higher than the DLS and increasing. So it does mean that the decoding power increasing across all the stage, okay? But for the DLS is the same, it's still increasing, but it's still lower, right? So far, it seems that the both region DMS and DLS, yes, sorry?
Yeah, I was wondering how many paths are there in total? So what would be, I guess, threshold for the decoding power?
The number of paths is 12. But so what
So it's one of the 12 would be just guessing the
no, he has almost to do it 2 times. This path per session. Is it your question?
No. Just the overall number of paths possible, which are possible to take, are 12.
Yes.
So it's 1 over 12.
Yeah.
The coin flip.
Oh yeah.
Okay. And the decoding power is for decoding of the correct path, or
no? We put all the paths together.
So it's decoding power of
predict do we have any relation between neuronal activity and the path that the animal is doing? So it does mean that during this session, at least the animal has to do 2 times this path, this specific path, and can we say that this activity is predicting this specific path? Is it clear? Okay. So what I was saying, yes. So DMS increasing this decoding power and DLS in parallel, but DMS is always higher. And interestingly, we can see that at stage 2, we have a significant difference between decoding power in DMS compared to DLS. So again, it seems that at stage 2, DMS would be more involved in decoding the path, right? So to summarize all the things, what we have found? We have found that DMS and DLS are both engaged throughout learning, and it can be quantified. Remember, we have more coding neuron in DMS for event, task related, for path, left and right, okay? And we have seen that the decoding power is always stronger in the DMS compared to the DLS during all the stages, suggesting that the DMS is more involved than the DLS in learning and performing a spatial alternation task, right? So secondly, the proportion of DMS coding neuron decrease, whereas the number of DLS coding neuron increase over time. Remember, I told you that the DMS task-related coding neuron are during the 3 stage higher than the DLS, and after all, the DLS seems to have more task-related coding neuron. So, something interesting. The DMS decreasing in terms of coding neuron, but the DMS is always higher in terms of decoding power. Okay. So, this is something new that could be interesting to discuss, is the idea that the dynamic of the both systems, DMS and DLS, is probably different. I'm explaining. For the DMS, you decrease the number of coding neuron, but the network is still more powerful in terms of decoding behavior, right? So could it be a reorganization in the network? Like, for example, less coding neuron, but a stronger synchronization, or the strength of the interaction probably is higher between neurons, could be an explanation. And the DLS has the number of coding neuron, which is linearly related to the decoding power, because we have seen that the coding neuron in the DLS increasing in the same time than the decoding power during the 4th stage. So to conclude, these results suggest that the DMS and DLS neural networks are engaged during all learning stage, but they're gradually reorganized in different way, in contrast with the common assumption of the gradual shift, because I didn't mention that at the beginning, that in the common literature, we think that the DMS is involved at the beginning doing the goal-directed behavior, and the DLS takes the relay. So this is not, I mean, in coherence with that we found for this type of behavior, which means free learning, if we can say it's a learning without any time step. Okay, so I conclude with this slide, and I would like to thank my team, my IPMC team, and a nice picture here in front of the sea. And I would like to thank all of you for your attention, and oops, I'm ready for questions.